grab some ice. Anything else?
No, I think.
Okay.
You can have one of mine. Go ahead and take one of mine.
Oh. Somebody could be right here. It's not, you're not in the way.
How's it going? Going all right. How are you? Good, not bad.
Thank you. Yeah, that's a nice big stuff.
Okay, good. Okay, so moving in.
Okay, so last time we did yeast transformation. This time we're going to be doing bacterial transformation. So let me kind of remind everybody what our project is, especially if you weren't here, then we'll kind of go over our results and what we're going to do today. So what we're doing is our project is in the field of what we call synthetic genomics. So making genes, making genomes from scratch. In other words, for chemically synthesized DNA that might not have ever existed previously. So rather than making a whole genome, which is huge, what we're doing is we're making what we call a model gene, which is GFP, or green fluorescent protein, which is a protein that's going to glow green in cells. It was originally isolated from a bioluminescent jellyfish, but now it's a gene that we can just use and manipulate in the lab. And so we initially, in week 1, made this GFP gene from small pieces of DNA that were about 60 base pairs long. And we had about 20 of these, and we put them all together. And all of these little pieces were synthesized in the lab chemically. We just told them what sequence of DNA we wanted, the A's, the T's, the C's, the G's. They sent us these small little pieces, and we assembled all those little pieces together until we got the full-length GFP gene, which is about 720 base pairs long. So then what we have to do in order to get GFP actually expressed in cells, because all we did was make GFP in a test tube. So it was just a clear, colorless liquid. And without a cell to actually get this DNA, remember this is DNA, protein is what's actually going to glow in the cell. So we need a cell to take this DNA of GFP and convert it into RNA and then into protein to actually get things to glow. So that's what we've been doing in the past couple weeks. We've been doing the process that we call molecular cloning, which is to take this piece of clear, colorless DNA in a tube and to introduce it into cells so that those cells can then convert the DNA into RNA, into protein, and then the cells should glow green. Now we've been doing this by 2 different methods that we did last week. So in order to get this DNA into a cell won't just pick up a random piece of DNA and keep it. It only keeps what it needs. So in order for this piece of DNA to get into a cell and then be maintained or be kept in that cell, it needs a holder, and we call that a vector. The vector is just a piece of DNA that you join to your gene of interest. And that vector has some kind of a gene, a selectable marker. And the selectable marker is a gene that the cell needs to live. So the only way that the cell can live is if it keeps this piece of DNA, the vector and your gene, in the cell. So for yeast, our selectable marker was a gene called Hist3. It makes the amino acid histidine, which is one of the nutrients that yeast need to live. So yeast need this gene to be able to make one of their nutrients. So they have to keep this plasmid in order to live. Okay? For bacteria, the gene is what we call ampicillin resistance. So it makes the bacteria resistant to the antibiotic ampicillin. So when bacteria are forced to grow in the presence of ampicillin, they'll only grow if they have that gene. That gene allows them to essentially inactivate the ampicillin so they don't die. Any questions about that so far? Okay, so the question was, how are we going to join our gene and our vector together? And we did two different methods, and we really wanted to compare. And next week we'll be able to compare the efficiency, because we'll be transforming them both into bacteria today, and we'll be able to see side by side which one worked better. So we used two different forms of joining the vector and the GFP gene, and that's Gibson assembly and yeast assembly. So the vector is actually double-stranded. We'll draw it as double-stranded. Now, the way that they both work is that your vector and your gene have to have the same sequence on the ends. So let me draw it better so that they're actually overlapping. So they have the same sequence at the end. And Gibson assembly uses enzymes just in a test tube, we call it in vitro, in the tube, to put the vector and the insert together. And it's a three-step process, but essentially what Gibson assembly does is it uses an enzyme, we would say, to chew back or to remove a portion of the DNA so that you have these little single-stranded ends. And we also need to chew back the gene so that it's also single-stranded. And then, remember, DNA is double-stranded because the two strands can base pair with each other. A's pair with T's, C's pair with G's. So because these are the same sequences, these single-stranded pieces are going to join up and base pair together. And then a third enzyme in this mix called ligase is going to seal them together to make one closed circle, which is going to be your GFP genes plus your vector together. So we did that last week, and our results are right here. It should be our GFP gene and our vector sealed together. Questions on that? So that's Gibson assembly. The alternate that we did is what we call yeast assembly. And in yeast assembly, there are no enzymes. So yeast very naturally do this process of taking overlapping DNA sequences and combining them. It's a process we call recombination. And every cell does it to some extent. Yeast just do it a lot with a much higher efficiency. So essentially all we do is we combine our vector plus our gene. We make sure that they overlap. And one of the differences here is that the overlaps in yeast tend to be longer than they are in Gibson assembly. In Gibson assembly, they only need to be about 20 nucleotides long. For yeast, they need to be about 60. But then the yeast will simply do recombination and combine those ends to create one closed circle. That's the vector plus the insert together. No enzymes needed. You simply introduce those into yeast. Any questions? Now the reason there are these two methods, Gibson assembly when it came out maybe 10 years ago initially, but it's really kind of caught on in the past couple years, is really excellent at putting DNA pieces together. But, you know, if you buy the kit, which is probably the most optimized way to do it, they say you can put together maybe 4 to 5 pieces of DNA this way. So in other words, if we wanted not only the GFP gene, but a couple other sequences as well, we could put them together by Gibson assembly. In contrast, yeast who love to do this process of recombination, you can get up to 25 different pieces together. So that's why we use both procedures. It really depends on what you want to do. If you only want to put 1 or 2 pieces together, go for Gibson assembly. It's nice and quick. If you want to put together a lot of different pieces of DNA, you're going to want to do yeast assembly.
That gives it a time difference.
The time, there's also a time difference. So this would be, Gibson assembly took us, what, it was 50 degrees for 15 minutes? And yeast assembly took us about an hour and a half to do it, plus till the colonies grow on a plate, about 2 days. So Ollie's right, if you really want something quick, if you just want to put your
All right,
vector
I'll
and
take a good
together,
movie
it's
for you.
definitely the way to go.
Sure.
Now, I should mention that I've been mentioning GFP for those of you who have been through this the whole way through. You know that in order for the
Take it easy, it's
GFP
okay.
gene to actually get made and expressed in cells,
You look
it
great,
needs
don't worry
another
about it.
sequence that tells the cell to actually take this gene and make protein. And that sequence is called a promoter.
Deep breaths.
Now, many of our vectors come with a promoter that just says, make this protein. All the time, any place, constantly make the protein. So for those of you who are just making GFP, it comes with a promoter that just says, make protein. Some of us are also putting together, instead of just GFP, we have a third piece, which is the RNR3 promoter. And that's a specialized promoter that says make this gene, make it into protein only when the cells have been damaged. Specifically only when the DNA has been damaged. So these cells should glow green only following DNA damage. So it's kind of a sensor of damage in the cell. Okay? So some people have put together just the 2, GFP and vector. Some people have put together 3, RNR3, GFP plus the vector. Just to kind of make things a little bit more fun and to give us a little bit of a more biologically relevant project. Okay, any kind of questions on what we're doing? We're all cool with that? Okay, so right now we've got these 2 constructs. The Gibson assembly is just DNA in a tube, no cells present. The yeast one is a yeast plasmid that's in a yeast cell. It's been made in a yeast cell. So it's going to be here as a colony on a plate. Okay. Now, to actually screen and verify that we have the construct that we want, that it's GFP in the correct vector, what we need is to get our DNA into bacteria. So what we're going to be doing today is putting the DNA into bacteria using a very standard procedure called bacterial transformation. And so this DNA that's in a tube is ready to go right into bacteria. This DNA, we first have to get it out of yeast so that we can put it into bacteria. So we have to do what we call plasmid extraction, where we take these yeast cells and we're going to open up the yeast cells, release the DNA back out into a tube, and then put it into bacteria.
Why would we do that? I mean, what's the reason?
Because it's much easier to screen them in bacteria to see which ones are correct. Once we have it in bacteria, we can make a permanent stock or store of them. The bacteria should also grow green, much more cleanly and easily than the yeast.
So if I were to damage the yeast by UV light or something, would it grow green, right? It was correct.
Theoretically, if you have the RNR3 and the GFP assembled correctly, yes. But anytime you do a transformation, what you're going to get on the plate is a mix of what you want and some things that didn't assemble correctly. A lot of times you're going to get just the vector without your gene in it at all. So that's why it's really important every time you assemble something, just because you have colonies on the plate doesn't mean they're actually the right thing. You always have to go through the screening step, which is what we're going to do next week.
So if I would put something like a penicillin or something,
Put a what?
something like penicillin or something resistant gene with this attached to it, would I get more pure colonies? With the promoter, I mean, if I add something one more piece that says, make it penicillin resistant or something.
Yeah, yeah.
And put it in yeast, would it make it more pure, the colonies more pure, or still would there be artifacts?
Well, it depends. If you're assembling it for the first time, you're going to have artifacts. If it's already a purified, closed circular plasma that you know is exactly what you want, and you put it into yeast, 99.9% of what you get is going to be correct. It's that assembly process of putting the pieces together that's very inefficient, so you always, every time you assemble, you have to verify.
Now, I have around, let's say we have around 5 or 6 different colonies in yeast or whatever, right? So how would I know which one is the right one? How would I not know? Oh, I just forgot everything.
If you have a bunch of colonies, how do you know which is the right one? You have to screen them. So that's what we're going to do next week. So you have to look at every colony and ask, is it the right one, and verify. Other questions? Okay, so
If we don't have any colonies on our experiment, what caused that likely?
okay, so here's, before we talk about how we do it, so we can evaluate your transformation. So this is why we always do a positive and a negative control every time we do any experiment. So your negative control, we put no DNA into the yeast cells, which means we expect no colonies, right? Because it shouldn't have that. It's 3 gene, nothing should grow. So first thing you always want to check is that your negative controller, NC, has nothing on it. If it does have colonies, it means you've got some kind of a contaminant, right? Either usually it's bacteria that was on your pipettes or something like that was just in the environment that contaminated your work. So first thing to check is that your negative control has nothing. Second thing to check is your positive control. So your positive control, like Ali just mentioned, is a plasmid that was already assembled. It's a closed circular plasmid that's histidine resistant. It should go into the yeast cells very efficiently and make a lot of colonies. So if your positive control gave you a lot of colonies, or some colonies, it tells you that your yeast transformation, the process of simply getting DNA into the yeast cells, worked well. If it didn't, it tells you that the process of getting DNA into the yeast was not very efficient. So that's the second thing you want to check. If your positive control looks good, your transformation worked, the last thing you want to check is your actual GFP plate, right? Your plate where you assembled all the pieces together. And there, if you have colonies, great. If you don't, it means that your assembly didn't work. So there you probably repeat again. Usually what we do is I probably add more DNA. If you're getting no colonies, I'd repeat the transformation maybe with 5 times as much DNA and see if you've got colonies. If your transformation didn't work, your yeast transformation, I just repeat the whole transformation process again until that works. So that's how you can tell, is the problem the transformation or is the problem the assembly? So that's why it's really important to have that control. Any questions? And usually because a lot of times, you simply forgot to add something or it's just it's not the experiment, but it's usually just a procedural mistake. Which always, this is first time through for everybody, right? So it always happens. So, did anybody's work? Does anybody have positive control? I mean, you won't call you need.
Yeah, the controls are both good.
The controls are good.
You got one.
Like this color?
Yeah.
Yep, that's a colony, yep. So,
You got yours.
now Victoria, yours were in the incubator a little bit less because until I played with them. So oh, those are gorgeous. Those, yeah, you've got a lot of them, right? You've got at least probably 2 dozen there. This one's just an air bubble, right? Yeah. That's a big circle, but that's really nice. They're just smaller than everybody's because they were in the incubator a little longer.
Okay.
So very nice.
Yeah.
I wasn't sure if that's what you were looking for.
That's
or if
exactly what you're looking for. As long as you don't have any on your negative control that look like that. Your negative control
Negative
should
control,
be
there's
clear.
no-
Yeah.
I know you're, I know you're
It's awful.
Yeah.
You've got tons of colonies?
Yeah, like,
Oh, that's mold.
Oh,
It's just mold.
yeah.
Yeah, your experiment is fine. So see that fluffiness? I saw it on a couple of plates. So the air is full of mold, you know. We don't like thinking about breathing it in, but it is. So those plates, you know, they're full of media, and especially for yeast, they're very rich in glucose. So if you leave the top on, you know, if you just leave the plate open, molds are gonna, you know, that's how polypenicillin was discovered, right? Because it just grew on a plate. So that's generally just a matter of either this lab being particularly molding or just technique. And one of the things that you learn is to only open the plates long enough to add what you need to add and keep them closed as much as possible to kind of keep down the mold. But that's just, everybody gets mold and there's nothing to worry about.
These two can buckets.
Hmm?
These two can.
Some people do, they take a Bunsen burner and keep it to kind of flame the area and keep the area around them sterile.
That's what we use. Burn a hand.
Yeah, exactly. So it's how comfortable you feel with just working next to flame.
So how do you go from stuff on here to putting it in the test tube? Because all it
Okay.
says is put it there.
Yeah.
Really explain how.
All right, so let's talk about the procedure. So the first thing that we got to do is we got to get our plasmid out of yeast.
it's a
Now,
brilliant lead-in.
With yeast, if you remember, is that yeast have both a cell membrane on the outside, which is, you know, just a lipid layer, but they also have a cell wall, which is really hard and really tough. So as fun as yeast are to work with, they're also a bit of a pain because they have that cell wall. So first thing that we need to do is we need to kind of strip off that cell wall from the yeast. So the first procedure you're going to do, very subtle, is you're going to take the yeast colonies and you're going to put them in a tube with some TE. TE is just buffer, and there should be a couple of tubes of it around in bottles that look like that. And then you just want to scrape the yeast off the plate. So this one. So if you have a couple nice big colonies, you just take a nice swipe off of it, and there's your yeast cells, and then you can just rub it off the side, and then vortex. Just hold it on to the vortex, and you'll see it'll become a nice smooth suspension. You want a vortex just until there's no clumps. Again, yeast have that nice cell wall, so you can be really rough with them, and they're fine. You can vortex the bejesus out of them, and they're okay. So that's an answer to your question. If you've got, yeah, vortex the bejesus out of them is a technical term, it's a specified length of time.
Jesus, 7.
If you have a lot of colonies like Victoria, what you can do, because you want all of them, right? Because some might be positive, some might be negative. You can just scrape like that across the plate and take that whole scraping and put it together into the tube. There you go. Any questions about that? Just with a toothpick, scrape it off. Then what we need to do is we need to get rid of that cell wall. So we actually do that with an enzyme called zymolase. Best names in biology. And it's simply an enzyme that chews the sugars in the yeast cell wall and essentially pokes holes in that rough yeast cell wall. So that should be on ice. It's labeled Zymo. So it's just, you add 50 microliters into your reaction. We put it at 37 degrees, and we let it go for half an hour, and we let it poke holes in the cell wall. You only need to be doing your GFP plate. You don't need to be doing your positive control. Okay? Once that's done, essentially now we can lyse the cells. So we're going to just use this Zippy Plasma Mini prep kit. I think it's with a Z today. Which is designed for bacteria, but essentially once you strip off that tough cell wall, you can treat yeast just like they were bacteria. And you're going to follow the kit. So essentially there's a lysis buffer, and this is always detergent to get rid of the lipids in the cell membrane, and it's also base, which helps poke holes in the cell membrane now. So you add that to your cells, you invert it to mix. It's very important that you mix your cells up very well. You add neutralization buffer, so this will take the pH back to neutral so you don't burn yourself when you work with it. And then you go through a series of column purifications. I know when we first made the GFPG, we put it over a column, right, to purify it. It's the same principle. You have a collection tube which is going to hold it, and then you have what we call our column, which is just a piece of plastic, and then that white is what we call the membrane. And that's kind of the working portion of the column. And essentially all it is that's your column, here's your membrane, and it's silica, which if you remember is positively charged. So when your double-stranded DNA comes through, which is negatively charged, it's going to bind and stick to the silica column. Okay? So all you're doing is crudely lysing the yeast cell. You're putting that whole mix over the silica column, and then as you spin it, again, we're going to spin it through the column, everything, all the garbage, the proteins, the detergents, the lipids are all going to flow through into the bottom of the tube, and the DNA is going to stick on the light membrane. Okay. Then it's got a series of washes. You can see step 9 and 10. There's endo wash buffer here, and then there's zippy wash buffer here, which essentially just gets rid of all the impurities. And then you add a dilution buffer, okay, which essentially, again, is going to pull your DNA off the column, again, just based on charge. And so it's going to be an aqueous environment that the DNA will resuspend itself in, and it'll come off the column. So what you'll wind up with at the end is a tube of purified plasma DNA.
And what keeps, how do we just get the plasma DNA back?
The yeast, okay, so right. So I drew this plasmid as being ginormous, but in fact, right, in a typical yeast cell, you've got 16 chromosomes in the nucleus, all matched up, and then you've got these small little plasmids out here in the cytoplasm, and these are what we want, the plasmids. In reality, if you wanted to purify the yeast DNA, and some of you I know have talked about projects involving yeast and making modifications to yeast genes, you would use a very, very similar protocol compared to the one we're using. The only difference is if you wanted the yeast genomic DNA is the way that you, so you do the same protocol as amylase to puncture the cell wall. When you take off the cell membrane, you're a little bit more gentle about it. When you use the particular detergents that you use are a little less harsh. But other than that, it's pretty much the same protocol. So even though we're purifying, what's coming off the column is plasmid DNA, there's going to every time be some of the yeast chromosomes in there as well. Okay? It's not going to be a problem because when we add it with the bacteria, the only thing the bacteria will pick up and maintain are the plasmids because they have ampicillin resistance. So even if any of these chromosomes get into bacteria, they don't have ampicillin resistance, so the bacteria aren't able to keep them. So that little bit of impurity isn't a problem. But every plasmid prep has genomic DNA. These come out of the cell because they're very small. These purify with a much higher efficiency than this.
So that's just going to stay wrapped up in the cell.
So essentially what you're doing is you're poking holes in the cell wall. So these just come flooding out of the cell. This is still in the nucleus, they're big, so they're much more difficult to get out. So it's an efficiency thing. So it's enriched for plasmid DNA with a bit of contaminated genomic. If you wanted the genomic, you would just shift the conditions to get more of that and to get rid of the plasmid. Yeah, big holes there. But gentle, you know, you sequentially strip off the layers. Yeah, yeah, but it's the same kind of a column purification. But you don't have to reduce the level. Any other questions? I mean, if you need help with walking through the steps of the columns, you can do it. Essentially, all the washes, everything flows through into the collection chute. The only thing that's important is when you do that last step, number 11, when you're finally going to purify your DNA off of the column and dilute it, you want to use a nice fresh tube, not one of these dirty collection tubes. That's the most important thing. All right, I will let you guys go to it. So you're scraping the cells into a tube, containing TE, adding Zymolyze, and then 30 minutes. If your stuff didn't work, you can either pick up, there's some plates here, actually there's just this one, or you can grab one of my plates that have GMP and use mine. If you're using one of my plates, you really only need cells from about half or a quarter of the plate because there's plenty there. Some of them.
Like the spin key, column this one, right?
Yep.
Into a clean,
That's this one.
and this one,
So you ready to spin?
and spin to one.
Yep.
So,
Okay, is this,
oh yeah.
is the collection tube the same as the Eppendorf tube? Or
Nope, which step are you in? You ready for
we're
it?
at the end of step 11, it says place the spin column into a fresh Eppendorf tube.
So that's going to be what's labeled the large 1.8 mil tube? Yep, that's going to be the outdoor tube. So the collection tube, the bottom now is just trash. Yep.
Yeah. No biggie.
We've still got the stuff, there's just lids everywhere.
Yeah. Was that a minute?
Probably not.